My project was on Alzheimer's disease, and before I go on to what my project was about, I just like to give a brief introduction on Alzheimer's disease. So for Alzheimer's disease, there's a few key hallmarks. One is something called amyloid plaques, which build up in the brain. And the way that those are formed is there's an amyloid precursor protein, and it's cleaved twice, and then one of the cleavages creates amyloid beta, and there are two types of amyloid beta 40 and amyloid beta 42. And they're both harmful, but amyloid beta 42 is a worse aggregate in the brain for Alzheimer's disease. And then another hallmark of Alzheimer's disease is the lack of acetylcholine and BFCN, which is basal forebrain cholinergic neurons. And so the basal forebrain cholinergic neurons help with the development and synthesis of acetylcholine. And acetylcholine is a neurotransmitter that's important for learning and memory, which makes sense because in Alzheimer's disease, learning and memory is impaired. And so what helps the BFCN is called BDNF, which is the basal-derived neurotrophic factor. And what that does is it attaches to a TrkB receptor, and the TrkB receptor activates and starts a signaling cascade that helps the BFCN survive. So you want to have the BDNF so the BFCN can stay focused. And in Alzheimer's disease, BFCN are very low. So it makes sense you want more BDNF. And so also amyloid beta accumulation is associated with lower amounts of BFCN. And so BDNF, the problem with it is that as a drug, it cannot pass through the blood-brain barrier, so therefore it is not a good therapeutic treatment to prolong the symptoms of Alzheimer's disease. So in my project, we use 7,8-DHF, which is dihydroflavone, which is basically the same thing as BDNF, except it's smaller, and so it still has that same high affinity to TrkB receptor. So our hypothesis was it would have the same effects and it would help offset the symptoms of Alzheimer's disease. So before I came in, what they had done was they had 4 groups of mice. 2 mice had the Alzheimer's disease gene in them, and 2 mice were wild types. And then 1 Alzheimer's disease treated mice group and 1 wild type group had the DHF protein. And so they were given at 1 to 2 months because we wanted to give them an early age, because in the real world application, you'd want to administer medicine in the early stages of Alzheimer's disease instead of later. And then they also put them in their water because ideally you'd want to be able to swallow the pill instead of inject it. And so they were given it at 2 months, and then at 3 months they were euthanized. And so I was given their right hippocampi, and in there I did 2 types of testing. One is called ELISAs. So what ELISAs are is you have basically a tray with different clear, almost like test tubes inside them, and you put each of your sample inside them, and then you have a system where there's a standard different type of concentrations, so you can find the different concentrations of the protein you're looking for. So in my experiment with the ELISAs, I found the different concentrations of amyloid beta and the different concentrations of BDNF. So basically, the darker you would look at it, and there would be different varieties of color, and the darker the color, the more concentration of that protein there would be. And the second type of method I used was Western blotting analysis, which is using gel electrophoresis. And so you take your samples and you put them through an electrical current, and the protein will separate based on their size. And so what we did is we would take it and put an antibody on it based on what we wanted to see in our results, because if we didn't put the antibody, way too much stuff would show up in our readings. And so then we would basically take this high-tech scanner, photo scanner, and we take it and we have these pictures of these different bands of protein. And what we were looking at, because we knew where the protein would be based on past scientific studies, and we look at the thickness, and the thicker it would be, the more concentration of the desired protein there would be. So that was my the results I did for about 4 weeks, and then after the 4 weeks of collecting data and doing the research, I collected my results. So right here was the APP protein and the amyloid beta protein, And there weren't any significant differences between the 5 fat mice, which were the mice affected by Alzheimer's disease, and the wild-type mice. And then the ELISAs showed no significant differences as well with the amyloid beta. And then over here, we tested TrkB, which is the receptor that BDNF attaches to. And so we tested 2 different types of TrkB. One was the full-length TrkB, and that's the one where we'll attach and initiate the signaling cascade. The other was the truncated TrkB, which the protein could still attach, but it doesn't initiate a signaling cascade. So essentially, it doesn't function. And so what we interestingly found, which we didn't find any correlation between other studies, was that the full-length TrkB didn't have an effect, but there was a higher increase of TrkB receptor of the truncated in the treated FAD mice. And so that was an interesting finding. So it could mean that the DHF protein is attached to the truncated part, and it wouldn't have an effect, the desired effect, if that makes sense. And so then with the ELISAs, we tested BDNF levels, and that was really interesting because the BDNF levels between the Alzheimer's mice, the ones that were treated with DHF, actually had an effect, and it increased the levels of BDNF. But among wild-type mice, the treated and untreated didn't have a statistical difference. And then over here, we tested gliosis, which is the amount of glial cells in the brain. And usually with Alzheimer's disease, mice that have Alzheimer's disease, there'll be more glial cells because they go into places where there's swelling in the brain. And the Alzheimer's disease mice both had significantly more gliosis than wild type, which makes sense because they're having more swelling in the brain. But the DHF didn't seem to have a statistically different effect. And the last thing we tested was neurogenesis, because in Alzheimer's disease, neurogenesis is very low. So that was one aspect where we thought DHF may have an effect, but it didn't really show anything. And so in a 6-week experiment, it's hard to come up with conclusive results because usually these type of experiments go on for like months or years and things like that. But what we found is that 2 reasons that we didn't find any conclusive results for DHF working is one, they put the DHF in the water, so that creates a variability when you're testing it because you don't know if the mice drank that day or how much they drank. So we suggested that in future experiments they do injections, so there's more a proper and measurable amount of DHF that's going into the mice. And the second is that these mice were younger, so they may have not had the same stages, like advanced stages of Alzheimer's disease, so their plaques and the deterioration of the brain might not be as significant. So another factor is testing mice that are more advanced stages of Alzheimer's disease. But I had a really good time doing the research. It was a lot of work, but it was really worth it. And I would recommend anyone who wants to do a research project to do it because it is a really rewarding experience. And I did mine at Boston University through the RISE program. And so I hope you enjoyed, and if there's any questions, I'd be happy to answer them.
How did you find the program?
Okay, so Hannah Weinstein in the year past, I don't know if y'all know her, but she had talked in the symposium about doing it, and the way it works is you can find it online and you apply. It's almost like kind of like applying to college because you have to send in your SAT scores, letter of recommendation, and you have to write an essay, and then you apply, I think you apply around now or to February, and then you usually find out in April or May, and they try to pair you with a professor they think they have space and that matches your interests.
There's a whole tile on the website of places that you can apply to, and this is the time to do it. So.
Would you be interested in like continuing this study and seeing like if it'll change, like you get more time?
Oh yeah, I definitely would because it's really interesting because it combines really a lot of aspects of neuroscience, and like this type of project has real world application because if we were to find something to help people with Alzheimer's disease, that would be really awesome. So definitely continue.